Okay.
Yes.
Hi, good afternoon. I'm Jim Hoxie. I am co-director of the Penn Institute for RNA Innovation at the University of Pennsylvania. And along with Drew Weissman, who could not be here today, I want to thank the RNA Collaborative for the invitation for us to present some of our science. Before I introduce our two speakers, I wanted to mention that before Drew and Catalin Carrico actually received their Nobel Prize in 2023 for their RNA work, this institute was created on our campus really to provide a home for the explosion of RNA research that was taking place in all kinds of areas. It's been a tremendously exciting process. Those of you, I think, have our website. You can take a look at it and see what's going on. But one of the things I wanted to mention is that in Drew's Nobel address that many of you have seen, you might recall that he only mentioned the COVID vaccine, the mRNA COVID vaccine, once. And he spent his time talking about the tremendously diverse number of uses that RNA science has, ushering in a new era of not only vaccines, but of therapeutics. So I wanted to show you you are well represented here. This is a look at the members of the collaborative seminar series. I want to remind you not to take pictures, or that this is being recorded and will be available on YouTube. And to just take a quick look, that'll be on the recording of other series that are coming up. So today, it's really a great pleasure for me to introduce two of our rising stars in the Institute. And their talks really reflect what I just said before, the breadth of RNA science. Neither of them are going to be talking about vaccines. Ted Kreider, who you will hear from first, will be talking about using RNA to deliver therapeutic antibodies. And then Jilian Melamed is going to be talking about very new approaches of using non-inflammatory RNA delivery systems to target autoimmune diseases. So you are in for a real treat hearing from both of these people. So with that, I will stop sharing and turn it over to Ted.
Thank you, Jim. So today I will be talking about some of our work applying the mRNA platform for in vivo expression of pathogen-specific antibodies. The impetus behind this work is actually in the part of my life where I'm an infectious disease doctor. And despite the explosion of biologics, particularly for cancer and autoimmune indications, we in infectious diseases don't have that many FDA-approved antibodies for infectious diseases. And they're listed here in black with the pathogen and the approved monoclonal. And you'll even note that for HIV, the only approved antibody is against a host protein, CD4. And there are a lot of reasons why this may be the case, but one crucial reason is illustrated by our experience with SARS-CoV-2, which is down here in gray because these are only available through EUA authorization. And that is due to the genetic diversity that viruses in particular exhibit. And so while we've had many antibodies against SARS-CoV-2, they're rapidly rendered ineffective due to the antigenic drift of the virus. And so pemivibart is really the only one that's currently EUA authorized that is virus specific. So whenever we have a problem, we ask, how can mRNA be used to address this problem? And so this audience is familiar with this, but I'll just go over it quickly. So depicted here is a schematic of an mRNA-LNP. And I like to think of an mRNA-LNP as having two really key components. The first is the nucleoside modified mRNA, which is shown here in the center in this black squiggle. And the reason why we really like this is because you can design your mRNA to express any protein you want. And then the second component is how you deliver it with a nanoparticle. And that's shown in the fall colors using a typical 4-component lipid nanoparticle akin to what is in the approved COVID-19 vaccines. And so we have applied this platform to many applications. I wrote too numerous to count here. People are aware of our vaccine immunogen designs, where we take vaccines to various pathogens and develop them. But also, we and other groups have been developing this platform for the delivery of host proteins to treat congenital diseases or to alter host cells as we would like to modify a disease state. I, as an infectious disease doctor, think about ways that we can use these to make tailored antivirals in work that I won't have time to talk about today. And we have a huge effort within our lab, and people around the world are applying mRNA gene therapy for immunotherapy to alter the adaptive immune system. And so I'll talk about one project or one application in which we can use this platform to make antibodies in vivo, essentially passively immunizing people without vaccination. And one of the first questions that I get asked every time when I talk about this topic is, how do you deliver mRNA-coded antibodies for expression in the circulation? And one way that we do this is we take advantage of a feature of lipid nanoparticles. And that is the fact that when you inject them intravenously, they go to the liver and the mRNA is expressed from the liver. To illustrate this point, here I'm showing you data from 5 mice that were injected with a luciferase mRNA-LNP intravenously and then treated with luciferin, the substrate for luciferase, 6 hours later. And you can see the luciferase signal as the heat map within this IVUS imaging. And the majority of the signal is found within the mice's abdomen. And when we dissect these mice, that signal localizes primarily to the liver. And extensive work within the field has shown that this is primarily through ApoE-mediated uptake by the liver, and that you can get liver expression of your cargo mRNAs. And we use this to our advantage because the liver is a highly synthetic organ. It makes proteins like albumin, protein S, and so on that get secreted into the bloodstream. And so if we provide mRNA that encodes a secreted antibody, the liver can essentially become an antibody factory for us. And so I'll show you data from a previously published study from 2017 that came out of Drew's lab, in which they designed an mRNA encoding an anti-HIV antibody called VRC01. And so in this experiment, mice were injected with one of 5 things: either luciferase mRNA-LNPs, the VRC01 protein, or VRC01 mRNA-LNPs at different doses. And then 24 hours later, they measured the amount of antibody within the serum of these mice. So the luciferase-treated mice had no VRC01 in their blood. Those that were given 600 micrograms of protein had about 100 micrograms per ml. And impressively, the mice that received 30 micrograms of mRNA-LNP had nearly double the amount of antibody within their blood. And they saw that when they decrease the dose, the amount of antibody that is produced by the liver decreases. And so when I joined Drew's lab, some lingering questions existed. So first, for diverse pathogens like HIV, we often will not make it with one antibody alone. We need to have multiple antibodies. And so one of the questions was, can we develop the system to deliver multiple antibodies at once? And then also, we want to match the antibodies to whatever model system we're testing. So could we alter antibodies to match the species that we're testing them in? So to test this, I took another HIV antibody, PGDM1400, and I made 4 different forms of the mRNA construct. First, to express an antibody, you have to express a heavy chain and a light chain. So we could encode that on 2 separate RNAs and mix them together, or make use of a bicistronic format, where you have one protein encoded, followed by a P2A sequence, followed by the second protein. And what happens is, as the ribosome translates this, it translates the P2A peptide, and when it reaches the last amino acid of the P2A sequence, it can't form a peptidyl bond. And so essentially, you get the generation of both the first cistron and the second cistron, as they're called, and you can encode 2 proteins from a single mRNA. And so then I designed these 2 mRNA or bicistronic constructs either to have the native human constant region, shown here in orange, or the rhesus macaque IgG1 constant regions, shown here in purple and pink, to see if we could match it to rhesus, which is an important model for HIV work. And throughout the talk, I will depict my antibodies this way. And the way that you can tell if something was expressed from two RNAs is it looks like a typical antibody. And if it was expressed from a bicistronic antibody RNA, you'll see a little black squiggle because that's the P2A peptide that's coming off of the first cistron. So we tested whether or not these would express within mice. mRNA-LNPs were injected, and then the mice were bled 1 and 6 days later. And then their serum was run on a denaturing gel and blotted with anti-human IgG for a Western blot. And encouragingly, in all of the mice that received our antibody mRNA-LNPs, we saw two bands consistent with a heavy chain and a light chain. But in none of our control mice, we saw any bands. And importantly, in our mice that received the bicistronic construct, the heavy chain band ran a little bit bigger than the one with the two RNA constructs. And that's consistent with the fact that this is a longer protein and it has the P2A peptide. So next we asked, what was the function of these antibodies? And one way you can measure function of HIV antibodies is using neutralization assays. So different HIV strains are incubated with the mouse serum at different dilutions. And we identify what is the dilution of the serum that 50% of the virus is neutralized, or the ID50. Every column is a different mouse, and every row is a different virus. So our top one is our negative control, and then we have 5 HIV strains. So the published pattern for this antibody is shown on the far right. That's always our comparator. And no matter which format we injected with, rhesus, 2RNA, bicistronic, or human, we saw the exact same pattern of neutralization that completely matched what was published. And so we wanted to ask if we could deliver 2 antibodies simultaneously and see the same thing. So in this case, I delivered half the amount of RNA of the anti-HIV antibody and an anti-flu antibody. And you can see that the titers dropped, and this is consistent with the mice getting half the amount of the anti-HIV mRNA. But we still have the same pattern of neutralization, and it's largely consistent in magnitude to a control where the other RNA was a reporter RNA, not a different antibody. And when we statistically compare these two, they are non-statistically different. We then tested the limits of this, saying, could we deliver 3 antibodies? So deliver an HIV antibody, a flu antibody, and a SARS-CoV-2 antibody. And we still see the same pattern of HIV neutralization in the mice that received 3 antibodies simultaneously. And so this suggested to us that we could just deliver all of the antibodies at once and potentially get the benefit of expression of multiple anti-HIV antibodies. So we designed an experiment to test that. So in this experiment, we have a subset of mice that received that same antibody, PGDM1400, that I just showed you all the data on. And I designed another antibody mRNA, PGT121. The details don't matter, but this targets a different part of the virus. So you've got 2 complementary antibodies. And on the right, again, you have the published patterns. And you can see because they target different parts of the virus, they neutralize HIV differently, where some strains are resistant to one, but other strains are resistant to the other. And the mouse data exactly aligned with what we saw within the literature. So our antibodies were properly expressed and their titers were as expected. When we treated mice either with the 2RNA format or the bicistronic format, we saw that the resultant serum was as if you added the 2 neutralization patterns together. So for instance, this virus, CNE55, which is sensitive to this one but resistant to the other, was neutralized by all the combo-treated mice, and vice versa with 398F1. And thus we can conclude we can actually deliver multiple antibodies against the same pathogen and see good activity of both. Now, I showed you on the last slide that there were some mice that got antibodies against 3 different pathogens. And so we had to take those sera and see if they could neutralize HIV, SARS-CoV-2, and influenza. So here are those 5 mice in which we performed neutralization against those 3 different pathogens. And we saw titers with ID50s of greater than 500 against all of these different pathogens, suggesting that these 3 antibodies were properly expressed. And while mice will not see HIV-1, SARS-CoV-2, or flu and get sick, these types of titers are the ones that if we could achieve this in a human, this could potentially have some benefit. So we're really excited about the concept of delivering multiple antibodies at once using mRNA. But this is an incredibly flexible platform that allows us to test concepts in the preclinical setting really, really easily. And so to illustrate that point, I wanted to switch gears and talk about another story that we've been working on. Now we're talking about a different pathogen called herpes simplex virus. Herpes simplex virus, of which there's one and two different viruses, is a large DNA virus. And unlike HIV that has a single surface protein, it has 11 surface proteins, 4 of which are implicated in entry and fusion. And so working with collaborators here at Penn, we identified 2 of those 4 entry proteins, GD shown in green and GB shown in blue, that we could identify neutralizing antibodies against. And those will be shown as the blue antibody and the green antibody from now on. And so I designed mRNAs that encode these various antibodies. For the green antibody, we use the bicistronic format, so it has a P2A, and you see the little black squiggle at the end of the heavy chain. But for the blue antibody, we used a different setup, where we have a single chain variable fragment followed by an FC region. And this means that this is shorter and thus a smaller antibody and is all expressed as a single protein. Now, to make our lives harder, we also wanted to see if we could favor bispecific formation or have the association of one arm of the antibody that is green and one arm that is blue. So to do this, we incorporated electrostatic steering mutations that are indicated on the slide. These are a type of knob and hole engineering. And the concept behind this is that if you express 2 different arms of an antibody, they may randomly assort where sometimes 2 blues go together, sometimes 2 greens, and sometimes a blue and a green, which is a bispecific. But if you create a knob and a hole where they fit together like a puzzle piece, you could potentially disfavor the formation of the monospecific antibodies and favor the formation of the bispecifics. And this has been well studied in protein engineering, but we really wanted to see, could we translate this to in vivo antibody production from mRNA? So in this experiment, we took all of our mRNAs, transfected them into HEC293 T cells, and harvested the supinatins to see what antibodies were present in the soups. And this, instead of, as compared to the last Western I showed you, is actually a native page gel. So we do not have heavy and light chains. We're instead looking at the full antibody. And so this green one runs quite large at around 180 kilodaltons, consistent with the fact that this is a full antibody with P2A peptides on it. As we expected, the blue antibody runs smaller because it's lacking both C1 of the heavy chain and the constant region of the light chain, and was highly expressing. When we mixed these 2 mRNAs together, we saw that there appeared to be 3 bands. So we see the small band consistent with the blue antibody, a large band consistent with the green, and if you squint, potentially a middle band here. And that could be a bispecific. We then tested this with our knob and hole constructs. And again, we saw a big band when we only delivered the green mRNA, a smaller band that runs a little differently with the knob and hole construct. It runs a little bigger, but 1 band here, and then a few bands here that were hard to discern, but maybe one of these is a bispecific. So we said, okay, let's move into mice. These antibody mRNAs look like they express. So we injected mice retroorbitally and then followed them for a month thereafter with serial bleeds. I'm showing you a native Western using blood from 2 days post-injection. So the first column is our luciferase-treated mice, and you see no bands. In our 2 mice that we selected to be representative here, the green antibody-treated mice, there is the large band, consistent with what we would expect. And in the 2 mice that received the blue antibody, we see a smaller band. When we mix them together, we see evidence of the small band, so there's probably blue antibody monospecific in there, but we also potentially could be having bispecifics formed. And when we do the knob and hole, you see that this smear is actually cleaned up, and we really have 2 bands. And we were actually really intrigued to see if one of these could be demonstrably the bispecific. But we could not assess that using Western blots definitively. So we had to develop an assay and ultimately turn to ELISAs in order to demonstrate that bispecifics were formed. So before we prove the bispecifics, we had to make sure that our ELISA works. So the way that this assay was developed is that the blue antigen was used to coat the ELISA plate, and then mouse serum or purified protein was allowed to bind to this blue protein. And so only those antibodies that have at least one arm, that is the blue antibody, will bind to the ELISA plate. Any green antibodies that are fully green won't. And so this first ELISA uses an anti-IgG detector. And so shown in the plot is the area under the curve from the ELISA assay, and each column is a different condition. So our luciferase-treated mice have no blue protein binding antibodies. Our green antibody-treated mice have no blue antigen binding titers. Our blue antibody-treated mice have resounding titers, which is good. So our assay seems to be working. And both of our mixture conditions, whether or not the knob and hole changes were present, have really high binding titers to GB, or the blue antigen. And our protein controls are shown on the far right. So the purified green antibody has no titers, and the purified blue antibody has super high titers. So now to assess whether or not these were bispecific antibodies, we changed the detector antibody. So now the detector, instead of recognizing IgG, is an anti-P2A antibody. So it will not recognize this fully blue antibody because there's no P2A peptide. It will only recognize those antibodies that are bispecific. And so when we repeated the assay with this different detector, we got a different result. So again, the luciferase-treated mice had nothing. The mice that received the green antibody have nothing because they don't bind the blue antigen on the bottom of the plate. The blue-treated antibodies, now they lack the P2A peptide, and so they gave no signal. When we mixed them together, with or without the knob and hole, we finally could detect signal. But there was a twofold increase in signal when we had the knob and hole changes. And we think that this is due to the fact that the knob and hole is favoring more bispecific formation. And then as our negative controls, the protein antibodies lack the P2A. So I'll show you some summary data. We have a lot of data on these mice, but I can only cover a little bit during the time allotted. So we can map what is the antibody level over time. So this is a blue antigen-specific antibody level followed over time and measured by endpoint titer on the y-axis. And so you can see only those mice that got either the blue antibody alone or a mixture of antibodies have detectable antibodies against the blue antigen. And we could even detect them out to day 28. So the mice that received the blue antibody alone or as mixtures still had detectable titers. Interestingly, our knob and hole treated mice, the titers waned faster, which is something we still need to follow up. We can also test whether or not there is a consequence of having these antibodies that protects the mice. So we designed an experiment in which we performed a high-dose genital HSV2 challenge on day 2 after treatment and compared treatment with a sham RNA, 1 antibody, or 2 antibodies. And we found that all of our negative control mice that received luciferase had clinical disease after virus challenge, but none of our treated mice did, suggesting that there was a protective effect to having neutralizing antibodies at the time of transmission. We also could assess whether or not there was virus replication as measured by plaque-forming units following swab of the mice. And we found that there was replicating virus in all of the negative control mice and 1 out of 5 of the green antibody-treated mice, but none of the remainder. And this viral challenge is actually lethal within mice. And so we could monitor for protection from death. So shown here in this Kaplan-Meier curve is our 5 groups, demonstrating that our luciferase-treated mice rapidly succumbed to their infection, whereas any of our antibody-treated mice could survive. So that was a bit of a whirlwind tour, but I hope that I was able to convince you that we can express combination monoclonal antibodies from mRNA-LNPs even when they're delivered simultaneously. We can exchange constant regions, and we saw no impact on Fab functions such as neutralization. And we think this moving forward will allow us to match the species that we are testing as a model organism relatively easily. And this may help us prolong how long the antibodies stick around and also prevent ADA induction, hopefully. We found that serum antibody levels were similar, whether or not we'd express them from two RNAs or one bicistronic RNA, and that we could form in vivo bispecifics using mRNA gene therapy. And so we think that this platform can be applied to any of a host of human health issues, including many other pathogens. And the way that we can apply this is basically however we can think. Maybe we can deepen our understanding of host pathogen dynamics. We do hope that this will eventually allow us to accelerate development for antibody-based interventions through the ease of mRNA production. So with that, I'd like to acknowledge the entire Weissman lab. Here's a picture from last year, as well as our funders. I wasn't able to talk about everything that I work on, but we also have a lot of other efforts. I do a lot of work to develop HIV vaccines, in addition to the antibody work I presented today, and the development of targeted antivirals. And then to highlight the people who directly contributed to this work, within the Weissman lab, Brianna and Justin work with me. They are research specialists who are currently on the interview trail for graduate school and combined degree programs. So please be on the lookout for them. I'd like to thank Drew, our collaborators at Acuitas, the Friedman and the Cohen lab, specifically Sita and Doina, without whom the HSV work would have never happened, Yu Hong within the Isaacs lab, and Shauna Shen at Duke University. And I would be happy to take questions.
Terrific, Ted. Thanks. So with the time that I'm allotted, I'm going to take the questions in the order they came in. First one is from Kate O'Neill. Very cool work, Ted, exclamation point. When you use multiple antibodies, do you see a more robust inflammatory host response? And could that limit the number you see, the number you use?
That's a fantastic question. So in general, we keep the dose the same. So the amount of lipid and the amount of RNA doesn't change based on the number of antibodies. So we have not seen any signs, overt signs, that we're inducing toxicity when we go between multiple versus one, but they're all receiving the same dose. We haven't measured more subtle markers of inflammation, such as inflammatory cytokines within the serum. But in general, it's the same dose across the board.
Okay, and there's a question, is there a difference in how long the antibodies last between single or multiple expressions?
Again, fantastic question. So this is one of the problems that we wanted to overcome with this study is, can we change the constant regions to match the host? And that is what we have found is the most important thing. So all of those data in mice using the HIV antibodies, those were human and rhesus antibodies that are rapidly cleared. They're gone around day 11, day 12. Whereas if we have the mouse antibodies, you saw that we could detect them out to day 28. So I think the biggest contributor there is the constant region and having these antibodies that can be recycled. But no, we haven't taken those studies out where we have multiple matched antibodies that we know have the same expression. Data I didn't get to show is that the green and the blue antibody, they express to different levels at the beginning. So they have different kinetics to start. The green ones clear faster. So lots of open questions.
Okay, and the questions are pouring in here. Somebody's going to pull the rug out from under us pretty soon, Ted, but what's the half-life of the blue and green antibody?
Yeah, so they differ and they have different starting concentrations. So in general, we think that it's a couple of weeks, akin to what's seen when a human antibody goes into a human based on the data that I showed you today. But more studies are needed to directly to measure exactly across different antibodies to see what it is.
And I think we have 1 minute. Do you load different mRNA encoding antibody in the same LNP? If so, do you assess each mRNA loading?
Those studies are ongoing. Measuring whether or not the same LNP has two RNAs for different proteins is incredibly difficult. So I will not be able to answer that in the time allotted. The answer is we can't. And in general, there's going to be random assortment.
Okay. And last one, this is the question I've asked you, Ted. So how soon after mRNA-LNP injection do you see antibodies in the circulation?
Every time we've gone closer, we've seen it. So we've done 6 hours after treatment and we see it. So they probably peak within the first 24 hours. It comes on really quickly.
And Ted, I don't know if you could put your ID infectious diseases hat on for a second, but tell us about herpes encephalitis and how antibodies like this might be useful in the clinical setting.
Yeah, so one of the frustrating things about being an ID doctor is a lot of infections, we don't have therapies, where we have one therapy that's been used for decades and is somewhat effective. So in the case of herpes encephalitis, which is a severe form of herpes simplex infection, we really have acyclovir. That's it. And the benefit goes down the longer it is until you get acyclovir on board. And morbidity and mortality is still high. So I view this as a disease where the stakes are high. We need to treat this severe infection, and people have serious sequelae afterwards. And so I think that it will be exciting to test these types of severe infections in animal models and potentially then in humans if there is any benefit. And that's what I mean by accelerating antibody-based discovery.
Yeah, and in this setting, the speed at which the antibodies actually get there is important. I think we need to move on. Ted, terrific, thank you. So shifting gears in a big way, we're going to go from using RNA to deliver antibodies to using RNA to target in a very different way autoimmune disorders. Jilian is leading a considerable effort in this project at our institute. And so Jilian Melamed, take it away.
Thank you for the introduction, Jim. Can you all see my slides?
Yeah.
Great. Well, I am so excited to share with you some results from studies that we've been doing to explore a relatively new area for vaccines that not many people think of when they think what sorts of diseases vaccines could be useful for. But today I'm looking forward to sharing my work developing tolerizing vaccines for autoimmune disease. And first, I wanted to just give a brief overview of what autoimmune diseases are and tell you how I think vaccines can be useful for curing or preventing them. In autoimmune disease, the immune system mistakes healthy tissue as foreign pathogens and destroys it. It's not completely clear what causes autoimmune disease, but one thing that is clear is genes that are involved in antigen presentation, the HLA genes, are among the strongest predicting factors for who will develop autoimmune disease. So in autoimmune disease, as I'm showing in the cartoon in the center of the slide here, dendritic cells can engulf and present autoantigen or self-antigen to lymphocytes. And then as a result of this, autoimmune tissue destruction can occur either due to T cell or B cell mediated processes where autoantibodies against autoantigens are often present and can mark tissue for destruction by the immune system. And in some diseases, tissue damage is also mediated by cytotoxic CD8s. So there are many known autoimmune diseases that affect a variety of tissues. Many are popular and you may have heard of them, such as type 1 diabetes or celiac disease, but some of them are more rare. And in aggregate, autoimmune disease can affect an estimate of 5 to 10% of the global population. And there are no cures for autoimmune disease. There are only treatments. So these treatments can include dietary and lifestyle changes, as well as systemic immunosuppression. And while this is very effective for managing some autoimmune diseases, one of the challenges with systemic immune suppression is that it can leave patients vulnerable to infection by viruses or even cancer. So we would like to take our mRNA-LNP tool and apply these to generate antigen-specific immune responses to prevent the development of autoimmune disease. And the reason we want to do this with mRNA-LNPs is because we know they're so effective at generating antigen-specific immune responses. However, they have lagged in their application to autoimmune disease compared to infectious disease. And that's because autoimmune diseases pose some really unique challenges that we have to overcome with the next generation of lipid nanoparticles. And the first challenge we need to overcome is that systemic delivery to immune cells in tissues that are relevant for autoimmune disease is incredibly challenging. And Ted and I did not plan to start our slides with the same experiment, but I will also share with you that when we intravenously inject mRNA lipid nanoparticles into mice or larger animal models, they induce protein expression primarily within the liver. And this is really useful for some applications, as Ted just shared with us, but creates challenges for others. This is for two reasons. There's some unique architecture in the liver that leads to protein expression specifically in that site. In particularly, the fenestrated endothelium unique to the liver leads to protein expression there. But also lipid nanoparticles can absorb serum proteins, which in turn bind to LDL receptors on hepatocytes. So here I'm showing you our favorite experiment where I'm showing you 5 different LNPs, and these are all considered gold standards in various different applications, formulated with mRNA encoding firefly luciferase. These LNPs were injected intravenously to mice and imaged by IVUS, and you can see that all of the signal occurs primarily within the liver. So how to achieve delivery to immune cells beyond the liver remains challenging. Another major problem we have when trying to develop mRNA-LNPs specifically for autoimmune disease is that LNPs are highly inflammatory. And this is extremely useful when we're developing vaccines for infectious diseases. We want that antigen-specific inflammatory response. But in autoimmune disease, that's what's already happening during the pathology. And what we know is that it is the lipid nanoparticle component that is specifically responsible for generating these pro-inflammatory antigen-specific responses. So here I'm showing you some data from a large collaborative group at Penn, where if we're comparing specifically the green dots, which are on the left, we have T follicular helper cells, and on the right, we have germinal center B cells that are developed in response to a protein antigen plus an empty LNP as the adjuvant. This achieves the same level of immunogenicity as the LNP containing mRNA encoding that protein adjuvant. So what we know from this data is that the LNP is acting as the adjuvant. The LNP is inducing the antigen-specific immune response. The bigger challenge in autoimmune disease is we also know that LNPs can exacerbate pre-existing inflammation, which is always present in autoimmune disease to some extent. So here I'm showing you results from a study by Hamid Paris, also at Penn, who generated a model of systemic inflammation and delivered intravenous mRNA LNPs to these mice. And if we look at the red squares on this graph of serum IL-6, we can see that when mice have pre-existing inflammation, that is exacerbated by the delivery of LNPs. So the big problem that we need to solve is we need to figure out how we can engineer immune-tropic lipid nanoparticles with reduced adjuvant activity. And to that, we're going to turn to the lipid nanoparticle composition itself. One component we're going to look at is the ionizable lipid, sometimes referred to as the lipidoid. And this composes about 50% of the lipid nanoparticle and has long since been considered the secret sauce that makes the LNP work. And that's because this lipid protonates at endosomal pH to facilitate mRNA delivery. And as we know from previous studies, this is the component that's largely responsible for adjuvant activity. So it's necessary for delivery, but we need to find a way to dampen this adjuvant activity to make LNPs useful for autoimmune disease. And to do that, I've turned to helper lipid components. LNPs also contain a mole fraction of helper phospholipid, and historically this has been thought to simply provide the LNP with structure and stability, and depending on the structure of the phospholipid, this can also contribute to endosomal escape. But over the past several years, there's a lot of really great work that's been done by several labs showing that helper lipids can be tuned to impart tropism for particular cells or tissues within the body. And the way that we know we can do this is we know from several studies that incorporating charged helper lipids into LNPs can reroute the delivery of liver-tropic LNPs to other organs. So here I'm showing you IVUS results from an ionizable lipid called MC3, which is incredibly liver-tropic. And what I want to call your attention to here is if we formulate these LNPs with a neutral helper lipid, DSPC, we have delivery to the liver. If we look at the middle panel and we formulate these LNPs with an anionic phospholipid, phosphatidylserine, we're seeing delivery to the spleen. If we incorporate cationic lipids, we can further shift the delivery to other organs. And this is a strategy that was widely popularized as selective organ targeting, or SORT, by Dan Siegwart, and much work developing this strategy has been published by Katie Whitehead's lab as well. But when I looked at this data and I saw these results, I was really inspired to consider what are the immunotherapy implications of these types of lipid nanoparticles, because as we know from our undergraduate chemistry and biology classes, when you change the structure, you change the function. So what I wanted to do was evaluate how these anionic lipids were able to shift not only the tropism of LNPs, but also to analyze what the resultant impacts were on adjuvant activity. So in these experiments, I generated a combinatorial library of 40 lipid nanoparticles containing GFP reporter mRNA. I delivered them intravenously to Black 6 mice. And then I used these mice to perform some serum cytokine analysis and to analyze splenocytes for protein expression and dendritic cell activation. So first, I would like to get everyone oriented to the kind of data I'm showing here. All of this data is going to be shown in a heat map format so that you can see the effects of all of the different LNP components I changed. On the vertical axis here, I tested 4 different ionizable lipids. SM102 and ALC315 are the vaccine ionizable lipids used in the Moderna and Pfizer COVID vaccines. 306-OI10 is a non-inflammatory ionizable lipid I helped develop while I was in Katie Whitehead's lab at Carnegie Mellon. And MC3 is a lipid that's commonly used for protein replacement applications and was actually the first ionizable lipid that was FDA approved for siRNA delivery. Across the horizontal axis, I'm going to show you several different LNP formulations that differ in their helper lipid composition. So right now, on the leftmost column in the graph, we're looking at mice that did not receive LNPs, and the heat map here is showing GFP expression in dendritic cells. If we formulate traditional LNPs with no anionic helper lipid, as we're looking at in this A column, we see very low delivery to dendritic cells. If we incorporate the anionic helper lipid DOPS at varying molar ratios, we start to see robust delivery to dendritic cells within the spleen. And we see varying levels of delivery to splenic dendritic cells with additional anionic helper lipids. So from this, I was really interested in determining what the impact on adjuvant activity was going to be. And when we talk about adjuvant activity, oftentimes we're showing T cell responses or B cell responses or protection from a disease. And those experiments are great, but they take weeks to months to generate enough data to draw conclusions. So the data I'm going to show you here is from a series of experiments where we looked at early markers of LNP adjuvant activity. And to get a good immune response for an infectious disease vaccine, something where you want an inflammatory immune response, 3 signals are required from our mRNA lipid nanoparticle. The first signal is antigen presentation, and we get that by delivering our mRNA encoding the antigen of interest. Quickly after delivering our mRNA-LNP, we get signals 2 and 3, where signal 2 is the upregulation of co-stimulatory molecules like CD80 and CD86 by dendritic cells. And the third signal required for a good vaccine response, a good pro-inflammatory vaccine response, is the upregulation of pro-inflammatory cytokines. So we can measure these things, and in an autoimmune disease application, what we really want to see is signal 1 preserved, but signals 2 and 3 depleted. So in our experiments, what we're looking for with our non-inflammatory lipid nanoparticles is signal one only. So just the delivery of our mRNA encoding our gene of interest. We're interested in seeing reductions in co-stimulatory molecules and reductions of pro-inflammatory cytokine production. And I will spoil it for you now. That is largely what we see. Here we're looking at the expression of CD86 and CD80 on splenic dendritic cells from mice that received our mRNA-LNPs. If we look at the column all the way over to the left, this represents untreated mice, so we're seeing low levels of these activation markers. When we deliver in column A, the formulation containing the neutral helper lipid only, so this is a traditional vaccine formulation, we see particularly with our vaccine LNPs, that SM102 and ALC315, that we have a sharp upregulation of activation markers on dendritic cells. And across the board, when we look at our LNPs containing our different anionic helper lipids, our DOPS, our BMP, and our PG, we see broad reductions in these activation markers. The same holds true if we look at the induction of serum cytokines in response to these LNPs, where with our vaccine formulations and also with MC3, we see an upregulation of serum IL-6, and through the incorporation of anionic helper lipids, we can reduce this. So all of these signs are good, that we have lipid nanoparticles that are going to be able to potently deliver mRNA to dendritic cells without inducing that pro-inflammatory adjuvant activity. So next, what we really wanted to know is this enough to elicit a tolerogenic immune response in a model of autoimmune disease? And the autoimmune disease I'm going to be talking to you about today is multiple sclerosis. And this is a chronic autoimmune disease that affects the central nervous system. In MS, the immune system attacks myelin, which is a lipid sheath on oligodendrocytes that insulates nerve cells to transmit action potentials along axons. And as MS progresses, this manifests differently in different people, but it can result in difficulty walking, in vision problems, in muscle stiffness, or in numbness or weakness in the arms or legs. And there are a number of autoantigens that are implicated in multiple sclerosis. But in humans, some of the best studied proteins are all structural proteins that are part of the myelin. And these can include myelin basic protein, proteolipid protein, and myelin-associated basic oligodendrocytic protein, or MOG. And MOG is the protein we're going to be focusing on delivering with our tolerizing vaccine in this study. So we were interested in testing, well, can we take our non-inflammatory LNP, formulate it with mRNA encoding MOG, our multiple sclerosis related autoantigen, and prevent disease development in a mouse model of MS. So here we turned to a model of experimental autoimmune encephalomyelitis, or EAE. And how this model works is black 6 mice are induced on the first 2 days using subcutaneous MOG peptide emulsified in adjuvant. And these mice also receive intraperitoneal pertussis toxin. So this is going to generate an inflammatory response against MOG in these wild-type black 6 mice. And the pertussis toxin is going to permeate the blood-brain barrier so that immune cells can get in and attack the myelin. And then on days 7 and 10 of this model, we treated mice with our non-inflammatory LNPs containing MOG mRNA. We gave these mice 1 microgram through an IV administration. And the controls we used in this experiment were either PBS injections or mRNA LNPs containing an irrelevant antigen, in this case, luciferase. And then for the remainder of the experiment, we monitored disease severity in these mice. To give you an idea of how we assess this model, this is EAE is a progressive paralysis model. So a number of days after EAE induction, the mice will start to exhibit signs of paralysis that we can measure by behavioral scoring. So first the tail will start to go limp, and then the hind limbs and the front limbs will slowly become paralyzed over the course of this experiment. Here we're looking at data representing our PBS control mice. So we see that mice develop EAE symptoms at around day 12 or 13 of the model. This peaks at around day 16. And mice, unlike humans, are capable of remyelination. So after the disease peak, we do see some recovery. If we look at our LNP treated groups, we see that our mice receiving luciferase containing LNPs develop disease pretty much similarly to the PBS control, whereas only in the MOG LNP treated group, we do see a stark reduction in EAE severity compared to our controls. So this is relatively new data, and we still have a lot of work to do to fully understand this mechanism that we're seeing. So ongoing work will include histology to look at damage to the myelin and whether or not our MOG LNPs can prevent that, as well as doing some work to really understand the cellular responses that give us this result. But what I hope I've convinced you of today is that LNP lipid composition is a really powerful tool that we can leverage to induce the kind of immune responses that we want to see. We can engineer LNPs to induce potent antigen-specific inflammatory responses and also potent tolerogenic responses. We know from this work that anion-containing LNPs can elicit antigen-specific immune tolerance. And we're looking forward to continuing to understand the cellular responses to LNPs in this model, as well as applying this tool to other models of autoimmune disease. So with that, I would like to thank my lab for their support of this work. In particular, within the Weissman lab, this project has been supported tremendously by Jenna and Michael, as well as Leslie, for lending her EAE and neuro expertise to all of this. We've been supported tremendously by Hung Sun, who has expertise at Penn in multiple sclerosis, as well as all of our funding sources for supporting this work. And thank you so much for making time to come listen to us share about our research today.
Thanks, Jillian. So the Q&A box is open, and while people are thinking of questions to put in the box, I'll kick it off, Jillian, with a question for you. So you showed us with the experimental MS model EAE. So those are animals that are normal, and then they have an insult that leads to a model that looks like MS. So there are many autoimmune diseases, and these interventions you're thinking about are going to be targeting autoimmune problems that already exist. So they're established. And so the question, could you take us maybe to the autoimmune disease of your choice to give us an idea how in an already established autoimmune disease, how you would think about what to target, what the tropism is, just take us to another example of how autoimmunity as a broad area could be targeted.
That's a really great question. And when I get this question, my answer is always the same. It's going to be different for every autoimmune disease. Every autoimmune disease is characterized by different patterns of inflammation. And that is what we found is that always changes the delivery of our LNPs. So whether that involves changing the route of LNP administration or adjusting the formulation slightly or tuning the dose, I don't think there's going to be one LNP solution that fits all autoimmune diseases. We know that this is going to be good for autoimmune diseases that are driven by a single antigen or a handful of antigens. Which antigens end up going into the vaccine are likely going to they'll depend on the autoimmune disease of interest, and they'll also depend on the specific patients.
Okay, so the logjam is released. So great talk, Jillian. Do you observe any increased inflammatory cytokines or death after injecting the MOG LNP?
We don't observe any increased cytokines in response to the MOG LNP specifically, but the biggest challenge with this work, as I kind of hinted at it at the beginning, is whenever you have pre-existing inflammation, you are going to be exacerbating an immune response. So much of the challenge of this work was fine-tuning the LNP formulation and the dose and the route of administration to prevent acute inflammatory responses to the LNP. We do see that this LNP formulation is tolerable to these mice, and the size of the LNP is a critical factor in determining what's going to be tolerable to mice in autoimmune disease models.
And the second question is sort of getting at I think similar to the question I asked. You started the injection prior to the injury, showed any score. Sorry, you started the injection prior to the injury, showed any score at day 7. Therefore, this intervention is prophylactic. Can you talk about it?
Yeah, absolutely. This is a prophylactic intervention that certainly has utility in the clinic. We know what patients are more susceptible to autoimmune diseases like multiple sclerosis based on mutations in the HLA genes. So there's certainly utility for a prophylactic like this in the clinic. In future work, we would like to explore dosing mice that have already started scoring or showing signs of already established disease. And I'm sure that will come with more challenges. But there's only one way to find out if this will be useful as a therapeutic intervention as well.
Okay. Next question from Allison Christiansen. Have you compared inflammatory versus monitory LNPs in EAE? Inflammatory versus non-inflammatory LNPs?
Yes, we have compared inflammatory versus non-inflammatory LNPs in EAE. Inflammatory LNPs are toxic to EAE models.
Okay. Timur Yarovinsky, Jilian, very nice and clear presentation with promising data in the EAE model. Quick comment. Having the tolerogenic LNP expressing the payload is not bad, as liver itself is a tolerogenic organ.
Yeah, absolutely. That could very well be working in our favor here.
Next question. Can you treat once the B cells are already forming pseudo lymph nodes in the brain?
We haven't tested that specifically. We will have to do those studies and find out.
Okay. Next question. Interesting comment on size effect on tolerable LNP. Is big or small LNP more safe? So
Small
size of the
LNPs are more safe.
okay. Also, can you add a checkpoint inhibitor into the payload, PD-1, L1, or mRNA? So
Have you been reading my lab notebook? Do you know what kinds of studies I'm planning to do next? Yes, I hope so.
all right. Pronay Roy, are these LMPs immune cell specific? Like we know how SLE is highly aggravated due to dendritic cell activation, specifically plasmacytoid dendritic cells.
Yes, these LNPs are immune cell specific. The immune cell populations that they target seems to change with different disease models, and I don't have enough data to say much more other than that right now.
Okay, Lisa Tan, what would be some examples of off-target effects with LNP mRNA?
One example of an off-target effect that we wouldn't want with a tolerizing vaccine is if you run the risk of inducing too much bystander tolerance, which manifests as systemic immunosuppression. So you would want to avoid the scenario where you are inducing a tolerizing response broadly to too many antigens. You want the immune system to remain as intact as possible against pathogens.
Okay. And Kate O'Neill, exciting, Jillian. How specific is the inflammatory response to LMP to the species? So species-specific issues. If minimal response in mouse, does that translate to human? So it's getting beyond the mouse and to the animal of your choice. What do you think?
Kate, what a tough question. Um, we haven't tested these LNPs in larger animal models of autoimmune disease. Um, We have tested them in SHIV-infected macaques, and we find that the delivery profile to immune cells remains the same, and that the LNPs are tolerable. But we have not studied specifically the inflammatory response.
All right, and we're in our last minute, and so the last question, is it possible that injection of mRNA-LNP vaccines could tolerize rather than active antigen-specific immune responses, rather than activate antigen-specific immune responses?
Yeah, of course. It requires a lot of LNP engineering since current LNPs are inherently pro-inflammatory, but there are a lot of steps we can take to modify the lipid composition and alter that.
Okay, and I think that brings us to the hour.